All right, answer key video for the practiced assessment. So the first thing that you're going to be asked to do on the actual summative is to label a model showing transcription and translation. So let's check to make sure we have these correct. So first, I labeled these two processes. So transcription, this showing this process here. I know this because it's happening in the nucleus. This little enzyme here is reading the DNA and creating an mRNA strand. And then out here, this whole process is translation. So I labeled that right here. And then looking at the individual parts, so I labeled this strand right here, the original DNA. I know it's kind of difficult to tell because it's in black and white. And this molecule right here represents the RNA polymerase, which is the enzyme that reads the DNA strand and pairs up free-floating nucleotides to create the RNA, the mRNA. And then I labeled this guy right here, the mRNA. So a few different ways that you could have labeled that. You also could have labeled this one, or you could have labeled this one coming out of the nucleus mRNA. And then I labeled this the nucleus right here. This is the nuclear pore, so this is where the mRNA leaves the nucleus and goes out to the cytoplasm. And then I labeled these amino acids. These are just floating around in the cytoplasm, waiting to be paired up with a tRNA. This is our tRNA molecule right here, which means this is the anticodon, these three base pairs on the bottom of the tRNA. That is complementary to the codon on the mRNA. And then here is another little amino acid attached to the tRNA that is specific to this anticodon. And then I labeled this unit right here the ribosome, or the ribosomal unit. And these are also little tRNA strands right here. You also could have labeled this part the anticodon. And then this section right here, these are the codons, because remember, a codon is a set of three base pairs that codes for a specific amino acid. So you could label this one, this one, this one, this one, this one, or that would be the anticodon over here. And then this right here is the growing protein chain. These amino acids are being put together with a polypeptide bond to create that protein. And for these two questions, I'm just going to flip back over to the slides instead of writing these out. So you can hear me explain these steps. So on the summative, you also will have to write out the steps in transcription and translation. Remember, they're very similar. They both start with initiation and then elongation and termination. So the process gets started. Whatever the process is creating gets longer, and then the process stops. So I'm going to describe these one more time. So initiation, the DNA molecule will unwind, just like with DNA replication. But here the RNA polymerase, which is the enzyme that helps this process, binds to the promoter and then starts to build up that RNA strand. And the promoter, again, is a specific set of base pairs that tells the RNA polymerase where to start. Then the second step, elongation. So this is where the RNA polymerase just moves along the DNA strand and starts to attach those free-floating nucleotides to create this RNA strand. And the last step, termination. So when the RNA polymerase reaches a stop signal called the terminator site on the DNA, it releases the RNA, and then the RNA will then leave the nucleus to go find a ribosome. Like I said in class, if you feel like you need to watch this animation again, definitely go back and look at the animations I assigned in class. This animation right here on slide 22 is also excellent, and then this one that we watched in class is really good. Okay, so that was transcription, translation. Again, same steps, initiation, elongation, termination. Here's another good animation video to watch if you would like a little review. So starting with initiation, so here the tRNA attaches to a small ribosomal unit, which is this gray structure here, which then attaches to the mRNA, which is this strand right here. And then this ribosomal unit moves along the mRNA until it finds a start codon. Remember that magic combination of base pairs or nucleotides is AUG. Then the large ribosomal unit attaches. So that's initiation. Elongation, AUG, which is in the middle binding site here, binds to the right site. The amino acids then bind together and each tRNA moves to the next site. So again, I would definitely go back and look at an animation for this because it's hard to tell from this model, but these tRNA strands will move to the left as they read the mRNA. And then the first tRNA leaves the binding site, as you can see here, and then this one moves over, and then a new one attaches. And then as these tRNA molecules are moving in and moving out from left to right, they will start to build this amino acid chain that you can see down here. These will start to become linked together to create the protein. And then the last step, termination. So the tRNA will reach a stop codon, and there's 3 different stop codon combinations in the mRNA. And then a release factor, which is another protein, will bind to the codon and release the amino acid chain. And then everything breaks apart. So we have the mRNA here, which can be read again, the ribosomal unit, which can get reused, and the tRNA, which also get reused. And then this is our little amino acid chain, or our polypeptide protein. All right, jumping back over to the practice summative. So the next question says, below, explain how this model on the previous page showing how exons of DNA determine the structure. So remember, exons, that is the part of the DNA that's actually turned on and actually expresses a gene. The other part, the introns, those are like junk DNA, doesn't really code for anything. They determine the structure of protein because when protein is transcribed into mRNA and then translated into protein, with this process, whatever you started off with here is going to affect what you create here. So again, DNA is read and transcribed, written down into mRNA, which acts as a message to be translated into a protein. And then why are proteins important? What happens when the shape of a protein changes? So I said proteins are important for almost every function in your body. So we looked at examples of this: antibodies, cell communication, enzymes. And then when you change the amino acids in a protein, it will change the shape of the protein and how the protein folds. So remember that very first activity we did where you looked at an applet of a little amino acid chain, and you changed the identity of the amino acids, and it changed how it folded in on itself, because some amino acids are hydrophobic and some are hydrophilic. So that is a good animation for this question right here. And also remember, structure equals function. If you change the shape of the protein, you're going to mess with its function. Okay, and part 2. So the second standard is all about mutations. You will have to transcribe and translate an original sequence and then a mutated sequence. So you can check your answers here with me. So here's my original DNA strand. Here's my mRNA that I transcribed, and my 6 amino acids that I translated. And then for my mutated strand, it's this base pair right here. It should have been a G, but instead it's an A. So instead of becoming AAC, it now codes for AAU. But if you look at the amino acid that codon codes for, it's the same amino acid. So this would be a substitution. And there goes my phone. Substitution mutation, and a silent mutation because it codes for the same amino acid. And then explain how the mutation causes a domino effect. Well, you change the DNA, everything gets changed after that.